Hello everyone, welcome to the part 3 of dark reaction. In this video, we'll be studying the C4 cycle. In my previous videos, I've already discussed the C3 cycle and the photorespiration in part 1 and part 2 of dark reaction. If you have not studied that, please go to my channel and watch those videos. Let's start with the C4 cycle. The C4 cycle is called as C4 cycle because it's the first stable product that is formed in this cycle. And is a C4 is a 4-carbon acid that is called as a malic acid. Let's see what happens in this cycle in brief. In this cycle also, the fixation of the carbon dioxide occurs as it is a part of the dark reaction in photosynthesis, in which the carbon dioxide is fixed to produce the sugars. Here, the first acceptor or molecule of this carbon dioxide will be the phosphoenol pyruvate. When the carbon dioxide enters the C4 cycle, the C4 cycle mainly occurs in the two cells, that is the mesophyll cells and the bundle sheath cells. The bundle sheath cells are present in mainly the monocot plants and monocot leaves, in which the bundle sheath cells are present around this around the vascular bundles, and the mesophyll cells surround the bundle sheath cells. This arrangement of the cells is called as a Kranz anatomy. That's the you can say the key feature of these C4 plants. In the C4 plants, first of all, the carbon dioxide is picked up by the mesophyll cells. The mesophyll cells convert the carbon dioxide into the bicarbonate ion. The phosphoenol pyruvate picks the carbon dioxide as a bicarbonate ion and fixes into C4 acid, which is first of all the oxalic acid, which is the unstable product. Then it is converted into the malic acid. Then this C4 acid is transported to the bundle sheath cells by the plasmodesmata connections. After that, it undergoes the decarboxylation and forming the C3 acid. The C3 acid is again transported to the bundle sheath cells. In the bundle sheath cells, this C3 acid will undergo will regenerate the phosphoenol pyruvate. Let's see how these steps occur and what are the major steps of this C4 reaction. So, the first step of the C4 reaction is the initial fixation, where the carbon dioxide is fixed into C4 acid, C4 unstable acid, oxalic acid, which is transferred into the malic acid. Afterwards, the transportation occurs. The transportation occurs here in the two steps. First of all, C4 acid is transported into the bundle sheath cell. Then, after decarboxylation, the C3 acid is again transported to the mesophyll cells, where it undergoes the regeneration to form the phosphoenol pyruvate receptor molecule again. The final fixation occurs when the decarboxylation occurs. After decarboxylation, this carbon dioxide molecule in the bundle sheath cell will enter the Calvin cycle. In the Calvin cycle, the C4 plants perform this Calvin cycle to avoid the photorespiration. That's a wasteful process, wasting the carbon atoms. After that, the C3 acid formed is transported to the mesophyll cell, where it undergoes the regeneration to form the phosphoenol pyruvate. The C4 cycle is also called as the hatch and slack cycle because it was was discovered and invented by the Hatch and Slack. It is also called as the Hatch, Slack, and concentric pathway. The concentric was also the female scientist which gave this pathway independently, and this pathway is also called as H, S, and K pathway from the initials of all the three scientists. There are several plants in which these C4 cycle is occurring. There are some monocot and dicot families, mainly the Gramineaceae family, the grass family, having the examples of maize. In the maize, sorghum, sugarcane, and panicum, the C4 cycle is occurring. Several plants of the Euphorbiaceae family, Cyperaceae, Chenopodiaceae, Amantaceae, and Compositaceae family, also the C4 cycle is occurring. Let's proceed forward towards the next slide. Here also this C4 cycle is explained in form of the carbon atoms, is shown as the moles. Here, let's see. The carbon dioxide is accepted by the phosphoenol pyruvate, that is a 3-carbon molecule. The 3 carbons from the phosphoenol pyruvate and 1 carbon from the carbon dioxide as a carbonate ion enters to form the 4-carbon oxaloacetic acid, which is a sterile product. It immediately converts into the malate or the aspartate, which is transported to the bundle sheath cell by the plasmodesmata connections. Here the ATP is used during the transport. After this, C4 acid or malic or aspartate acid undergo the decarboxylation. Here the final fixation of the carbon dioxide occurs. The carbon dioxide here enters the C3 cycle or the Kelvin cycle to avoid the photorespiration. After that, the 3 remaining carbons form the C3 acid. We can call it the oxalic C3 acid. This C3 acid is transported to the mesophyll cells, where it regenerates the phosphoenol pyruvate. Let's proceed forwards towards certain characteristics of the C4 cycle. The C4 cycle is a light independent reaction because it occurs in the mesophyll cells like the C3 plants, but the Kelvin cycle takes place in the bundle sheath cells to reduce the buildup of the oxygen. After that, the C4 plants are mainly the tropical plants. They live under the xerophytic conditions. The C4 plants have the special type of trans anatomy. It's a concentric arrangement of the mesophyll cells and the mesophyll tissue and the vascular bundles. Let's see this arrangement in this views of this a grass leaf. Here the vascular bundle is present. The vascular bundle is surrounded by the bundle sheath cells. These are these barrel shaped cells which are inflated. These bundle sheath cells are surrounded by the mesophyll cells, forming the concentric arrangement. This arrangement is called as the Kranz anatomy. Mainly the Kranz word means the wrath. The wrath word is used for some cover or you can say the cloth used to cover the dead bodies. The fourth feature of the C4 cycle is that it initiates in the chloroplast of this mesophyll cells and have a well-defined grana. The chloroplast of these mesophyll cells have the well-defined grana and having a special type of reticulum that is called as a peripheral reticulum. Let's study the C4 cycle in a little detail. As I've already told you that the carbon dioxide is accepted by this mesophyll cell in form, and here it gets converted into bicarbonate ion, which is accepted by the phosphoenol pyruvate, that's the acceptor of the carbon dioxide, and is converted into the oxaloacetic acid. From the oxaloacetic acid is converted into the malic acid and the aspartic acid. These are both are the C4 acids. From here, this C4 cycle has got its name. That's because malic acid is the C4 acid, which is the first stable product of this C4 cycle. This malic acid and aspartic acid gets transported to the bundle sheath cells. Here, the malic acid undergoes the decarboxylation, and this aspartic acid gets converted into oxaloacetic acid, and the oxaloacetic acid again undergoes the decarboxylation. The malic acid produce the pyruvate, which is the carbon molecule. The pyruvate undergoes the amination, forming the alanine, which is transported to this mesophyll cells. The mesophyll cells, in the mesophyll cells, the alanine gets converted into pyruvate. The oxaloacetic acid, which had undergone the decarboxylation, gets converted into the phosphoenol pyruvate, which is transported to this mesophyll cells. The pyruvate, which was produced from this alanine, undergoes the fossilization using the ATP molecule and forms the phosphoenol pyruvate, and hence the phosphoenol pyruvate is regenerated. I think we have discussed this C4 cycle in detail. Let's proceed forward towards written matter. Here, mainly this C4 cycle is divided into main three steps: initial fixation, final fixation, and the regeneration. We can also add the fourth step as the transport of this C3 and C4 acids. In the initial fixation, the carbon dioxide combines with the phosphoenol pyruvate, leading to the formation of C4 compound, that's the oxaloacetic acid, which was unstable. This is transaminated to the malic acid or the aspartic acid, which is transported to bundle sheath cells through the plasmodesmata. After that, the final fixation occurs. The malic acid is decarboxylated to pyruvic acid. Aspartic acid is deaminated to the alanine. The carbon dioxide where released is fixed into through the C4 cycle, or through the C3 cycle. There's a Kelvin cycle picked up by the RUBP, that is the second acceptor of this carbon dioxide in the C4 plants. After that, the regeneration of the phosphoenol pyruvate occurs. The pyruvic acid or the alanine formed in the bundle sheath cell return to this mesophyll cells, where the alanine is deaminated through the transamination reaction to form the pyruvic acid. The pyruvic acid is converted into phosphoenol pyruvate and inorganic phosphate with the help of the ATP. The C4 plants as a whole required 30 ATP and 12 NADPH molecules to synthesize the 1 glucose molecule from the 6 molecules of the carbon dioxide. That's all for today. Thank you. Please like, share and subscribe to my videos, and please mention in the comments if you want any special and specific topic to be discovered. Please subscribe to my channel. Thank you.